Do a screencast. This time I'll be addressing post-transcriptional modifications, and it's the first time I'm using my new pen here, so just bear with me about trying to maneuver my way around the screen here. So by the time we finish this, we should be able to describe and explain how eukaryotic cells will modify the primary transcript to get mRNAs, to get tRNAs, to get rRNAs, and then we'll talk about a couple of other RNA molecules there. And then also being able to describe and explain the roles played by each of the above in gene expression. Okay, so we looked at before how the primary RNA transcripts are going to be modified extensively, or we looked before about how we got those RNA transcripts, and now we're going to be extensively modifying them to get functional RNA molecules. So in a prokaryotic cell, you have the transcription process happening here, and you're going to then almost immediately get this mRNA. Sorry about that. This mRNA is going to be translated immediately here. In a eukaryotic cell, you get the transcription happening, but then you take this mRNA that's right here, and you're going to modify that mRNA molecule. What is not shown in this diagram is the other types of RNAs, right? The ribosomal RNA that's going to be there, and then any sort of tRNA, that will also play a role in the translation there. So let's take a look and see here what's going on in eukaryotic cells with the modification of the mRNA, or the messenger RNA. So the title up here, post-transcriptional modifications. In the mRNA, we're going to get a 5' cap added, we're going to get a poly A tail added, and then we're going to have introns removed. So there's three things that happen here. One is making this 5' cap, 2, putting a poly A tail on there, and then 3, removing introns and then splicing those exons together. So from transcription, we get this pre-mRNA molecule, and on the 5' end of that molecule, we're going to put this 5' cap, which is, you'll see it referred to as a modified GTP molecule. Suffice it to say that this 5' cap thing right here is going to help guide the mRNA out of the nucleus, okay, into the cytoplasm. So the 5' cap gets slapped on to the end of that, and of course there's going to be an enzyme that does that. And on the other end, on the 3' end, we put a poly A tail, and this is exactly what it sounds like, poly A. So you're talking about a couple hundreds of just A's added on to the end of that. Now these A's, of course, are going to be non-coding. They don't code for any sort of polypeptide or anything like that, or any amino acids, so they will not be translated. But this poly A tail plays a protective role. And once the mRNA gets out into the cytoplasm, protects it from hydrolytic enzymes. That are going to be degrading that mRNA out in the cytoplasm. Finally, the last thing that happens here is that this whole mRNA does not get expressed, and some of the sections of this mRNA will actually get removed. And so those sections that get removed are known as introns, and the way I remember this is that they are in the way and they need to get taken out. And then the exons here get kept, and the way I remember this one is that the exons are the ones that actually get expressed. Right? So the next slide will detail how this happens, but you're essentially going to take this intron out, you're going to remove this intron here, and then you're going to splice this exon together with this exon, and then splice this exon together with this exon here. So you see that you get the introns cut out, the exons then spliced together to get then the, this is referred to as the final mRNA transcript. And this is the one that will actually be translated. Okay. Do take a look at this this animation down here. Okay. So a little bit more about how those introns get removed. So in a little bit more detail here, the introns are removed, the exons spliced together via something called an snRNA. And a protein complex that is all called a spliceosome. So a spliceosome is a body that's going to be splicing. So what is this thing here? So this is a spliceosome here. Or sorry, this is going to be a part of a spliceosome called made up of these things called SNRPs. This is how we pronounce this, a SNRP. And this snRNA right here stands for small nuclear RNA. So there's a small nuclear RNA, and this is a single-stranded RNA molecule that was then folded. You see how it's all folded here. And then there are some other proteins associated with that SNRP, and then you have these other proteins that combine together here. So when these SNRPs and these other proteins come together, you get this thing called a spliceosome. Right? So this structure is the spliceosome, containing a SNRP here, and a SNRP here, and a SNRP here, and a SNRP here. So these SNRPs have little pieces of RNA that are going to be complementary to this single-stranded RNA that's here, and you get this hybridization forming right there. You get a hybridization forming right there, and then when those hydrogen bonds form, that helps to catalyze the reaction. So you're going to be, this spliceosome essentially then is breaking phosphodiester bond there, breaking a phosphodiester bond there, and then is able to remove this intron here, and then form a phosphodiester bond between this and this one here, linking those two exons together. Right? The animation might do a better job of that, so do click on that. Moving on to a different type of RNA here, forming the tRNAs. So these tRNAs are transfer RNAs, and we're going to talk about how these are folded and then charged. So these are linear. These tRNAs start off as linear. So here's a 5' end, here's the 3' end, but then it's folded into this sort of like hairpinny loop here, and then you get another hairpin loop here, and then you get a hairpin loop over here. Right? And so this is it in 2 dimensions. Here they're trying to show it to you in 3 dimensions, and the color codes match up right? Here, and then here's this hairpin loop down here. Right? And then this pink hairpin loop is sort of back in 3-dimensional space. So your author here goes so far as to just make this whole thing a cartoon, and so this is the cartoon version of what the tRNA looks like. The 2 business parts of this tRNA molecule are the anticodon area down here, and the anticodon area. And this anticodon is 3 nucleotides that are going to be complementary to a codon on the mRNA. Right? So this linear transcript is going to fold on itself. You see you get hydrogen bonding happening here and here and here, so you get this little double helix area, and then you get the anticodon area down here, 3 nucleotides. I know it just looks like 2 of them here. They don't show the 3rd one. There should be a 3rd one there as well. And then over here, your author shows the anticodon as this area down here. The other end of the molecule is where the amino acids are going to be attached. Okay, so these tRNAs, of course, stand for transfer, and you're going to transfer amino acids from the cytoplasm to a ribosome. So this is just another detail, further detail about the enzyme. This is the enzyme that is going to be taking an amino acid using ATP as an energy source, and then catalyzing the placement of this amino acid onto the tRNA right there, and then this enzyme can be recycled back up here to grab another one. So the tRNAs, there are different tRNAs for different amino acids, right? So if you have 20 amino acids, then you have 20 different tRNA molecules that have different anticodons to them. And again, you can sort of go backwards here. So if the codon is let's see, A, C, G, then the anticodon for the tRNA is going to be, well, the complement to that is going to be U, and then G and C. Okay, but now how do you know which amino acid is going to go on there? You would look at the codon. The codon would tell you which amino acid goes on there, not the anticodon. But if I gave you the anticodon, you could go backwards to get the codon and then tell it that way. But remember that codon table is just for the codons, not the anticodons. Finally, then we get to the RNA molecules, and these are components of ribosomes. And this candy cane looking structure up here, I want to point out that it started off as a linear transcript, and so there's the 5' end of that linear transcript, and then here's the 3' end, so it's not just like a little maze to get there. So I'm going to go here, and then it hairpin folds. All these red areas are indicating areas of hydrogen bonding, so this is a very complex folding mechanism or structure here. So you get the rRNA, the ribosomal RNA, combining with some other proteins, and you then form what is known as a ribosome. So this is a computer model of it down here. So the green here and the red here are parts of the rRNA, and then all of this blue and orangey components, these are other proteins that are associated with that. So when you look at these ribosomes, though, there are used typically a larger subunit and then a smaller subunit. And eukaryotic ribosomes are a little bit different than prokaryotic ribosomes, so their subunits are slightly different sizes. But suffice it to say that when you get these small and large subunits coming together to form a ribosome, that ribosome will form 3 different pockets. Here's the E site, and then there's the P site, and then there's the A site. And of course, this is your author's cartoon version of that. Right? And then here's sort of a computer-generated model of that, where you get these little pockets where the tRNAs are going to fit in there. Right? The 2009 Nobel Prize in Chemistry was awarded to researchers that discovered this and the enzymatic qualities of these ribosomes. Right? So that's actually an interesting little NPR interview if you want to listen to that. Go ahead and click on that. And again, so here are the ribosomes, and you can see them in an electron micrograph here associated with the endoplasmic reticulum. So this would be the rough ER, and this would be the membrane of the ER here, and then all these little dots here, all these little black dots, are the ribosomes themselves. Okay. So there are some that are bound right to the membrane there, and then there are free ribosomes there as well. So it turns out, though, that we have defined the gene then as segments of DNA that code for a polypeptide. It turns out, though, the more we look, that actually many of the genes, so like gene 3 over here, this gene holds the code for not necessarily a polypeptide, but for just an RNA piece right here. And these RNA pieces could be a tRNA, could be an rRNA, could be one of those SN RNA molecules. where these are all single strands that will fold and be functional. They provide some sort of function in their three-dimensional structure. So the fact that RNA molecules actually do things in the cell, and it's not all just proteins that are doing things, right? So, I mean, proteins definitely do perform a lot of functions in the cell, but we've found out that actually some of these RNA molecules themselves are functional, and it turns out that many of the genes in the genome code for RNA molecules and not necessarily always coding for a protein or a polypeptide. Okay. We've seen this table in another screencast here talking about messenger RNAs and then tRNAs, ribosomal RNAs, this primary transcript here, and then describing the snRNAs in a little bit more detail. Okay. So hopefully that will you'll be able to describe and explain how eukaryotic cells modify this primary transcript to get the mRNA, and again that is going to be three things, 1, 2, and 3 things there, with the 5' cap, the introns removed, and then the poly A tail. The tRNA molecules, how they are the anticodon part to them, and then where the amino acid is going to be added. And then the rRNA molecule, how it folds and associates with other proteins to form that E, P, and A sites. Right? And then we talked briefly about the spliceosomes and those snRNAs and their role in catalyzing the removal of the introns. Okay? So hopefully that is helpful, and if you have questions, bring them to class. Thanks for listening.